Now let us introduce to you our 3D cell culture products, prime surface multiple well plates that used in anti-cancer drug discovery and regenerative medicine research. Prime surface multiple well plates are ultra-low attachment plates for 3D cell culture with Sumitomo Bakelite surface processing technology. The plate surface is coated with superhydrophilic polymer, making it suitable for 3D culture such as cell spheroid formation. The video shows spheroid formation in HeLa cells. Cells begin to aggregate immediately after seeding and form round spheroids in about 24 hours. In general, spheroid size changes when the number of seedings changes. So spheroid size can be tuned according to the purpose of the experiment by changing the seeding density. Next, we will give you some highlights on the performance of prime surface. As shown in the figure, prime surface has very high rate of a singular spheroid formation per well. The formed spheroids can be used as is, for example for drug efficacy evaluation, cytotoxicity assay, invasion assay, and stem cell differentiation induction. The size of cell aggregates is known to influence stem cell differentiation efficacy, and spheroid circularity is important for gradient profiles in drug screening. By providing round and uniform spheroids, prime surface is a great tool for 3D culture to obtain highly reproducible data. Next, we will introduce the well shape of the prime surface plate. First is the slit well plate. The slit structure at the top of the wells makes it possible to remove media of all wells from one plate corner, making the time-consuming media change process easier and also reduces risk of damage to spheroids. Furthermore, the slit structure allows more media to be shared, and the frequency of media change can be reduced by increasing the amount of media compared to conventional products. This slide explains how to change media in a slit well plate. You can aspirate media of all wells slowly from one corner of the plate while tilting the plate slightly. For details of the media change, please refer to the linked video. Next is V-bottom plate. V-bottom plate, with its incline angle of the bottom, successfully supports spheroid formation and cell aggregation of the cell lines which exhibit low cohesive force. And prime surface V-bottom plate is widely used in human iPS cell research. From here, let us introduce some prime surface plate applications. First, here is an example of brain organoids generated from human iPS cells using prime surface V-bottom plate for self-organization. By generating uniform aggregates from human iPS cells, it is possible to reproducibly and efficiently perform organoid differentiation induction experiments. Next, we have three applications in anti-cancer drug research. The first one is the high-throughput analysis using prime surface 384 well white plate. Prime surface 384 well white plate enables high-throughput operation from spheroid formation to chemiluminescence measurement. It is a suitable assay format for anti-cancer drug screening. The second is the evaluation of drug efficacy of anti-cancer drugs using 3D culture. In 2D monolayer culture, the anti-cancer drug acts directly, so the drug effect appears even at low concentrations. On the other hand, 3D spheroid culture can mimic in vivo microenvironment and cells respond more accurately. Compared to 2D culture, 3D spheroid culture is more similar to in vivo environment and can create hypoxic conditions of cancer tissue as shown in the figure. When cells were exposed to the anti-cancer drug tirapazamine, which is a hypoxia-triggered DNA-damaging cytotoxic drug, stronger drug effect was shown in 3D spheroid culture. 3D culture using prime surface constructs hypoxic spheroid microenvironment which is more similar to in vivo for drug efficacy test. Prime surface can also be used in cancer cell invasion assays with combination of hydrogels. Here is an example with Well Biosciences Vitrogel Hydrogel Matrix. After spheroids were formed in prime surface plates, Vitrogel Hydrogel Matrix was added and covered by culture medium on top. Then cancer spheroids started invasion progress by day 1 of the culture. Vitrogel Hydrogel Matrix has multifunctional ligands, and the combination with spheroid formed from prime surface is an appropriate tool for invasion assay. Thank you for your kind attention.